Nearly everywhere we humans tread, the ground at our feet is patrolled by a predator almost as old as life on land itself, and whose story, one of the longest in the animal kingdom, has been left largely untold until now. There's something about centipedes that makes them a uniquely captivating sight, as they navigate their world, borne ever forward by the rhythmic motions of their many legs. Their long, sinuous bodies appear to flow over the land with grace and fluid smoothness so utterly distinct from the chaotic scuttling and crawling of the insects and spiders that share their domain. Today, over 3,000 species of centipede are known, from diminutive soil dwellers only a few millimeters long to 35-centimeter brutes that can take down snakes and pluck bats out of the air mid-flight. But among the most noteworthy things about these multi-legged hunters is not their range nor their diversity, but the sheer tenacity of their reign, for these are some of the land's most ancient inhabitants, and the story of their evolution now lies scattered across the planet as cryptic, tantalizing hints, some entombed in the earth since time immemorial, and others hidden amid the anatomy and genetics of centipedes yet living and breathing today. Our journey begins in a period called the Cambrian, which began around 540 million years ago, well over twice the age of the dinosaurs. The lands of the Cambrian were a parched and desolate realm, hostile to life in almost every way, but out to sea it was a very different story. Life had begun in the oceans over 3 billion years earlier still. For much of that time it remained simple, even microscopic, but eventually larger, more complex organisms appeared. And by the Cambrian, the seas were brimming with all manner of curious creatures, the likes of which our planet had never seen before. At a glance, many animals of the Cambrian appear so otherworldly as to defy classification. Yet this strange assembly was the cradle of the animal kingdom as we know it today, with early members of most of the major animal groups alive making their grand entrance around this time, and the most prevalent among these were the arthropods. Typified by their armoured bodies and jointed limbs, the arthropods are a near immeasurably diverse group that today makes up over 80% of animal species known. The arthropod family tree can in turn be divided into two major branches. On one side there are the chelicerates, which include spiders, scorpions and horseshoe crabs. On the other side are the mandibulates, which include crustaceans, insects, millipedes and centipedes. Today, mandibulates exhibit a near boundless array of wildly different forms, yet all are united by their shared namesake feature, modified limbs called mandibles, which in most mandibulates are used to chew food. Early examples of mandibulates are ubiquitous in Cambrian sediments. Among the most primitive are a group called the Suonatores. I've lived here too long. These elongated, multi-segmented creatures could grow to lengths of around 15 centimeters, and some are known from exceptionally well-preserved fossils, which can tell us a great deal about their anatomy, and in turn the early stages of mandibulate evolution. One of the most striking details apparent in some Suonatore fossils is the intricate preservation not just of the animal's external form, but even aspects of their internal anatomy, including the brain alongside the rest of the nervous system. And it was examination of the Suonatore brain that provided key evidence to indicate these creatures were primitive mandibulates. The Liuto cerebrum, the middle portion of the brain, connected to the antennae in the same characteristic manner seen in living mandibulates, and the organization of the nerves connecting the brain and the eyes were similarly reflective of mandibulate anatomy. Further parallels with modern mandibulates have been found when examining the animal's cardiovascular system, which, just like the nervous system, has been preserved in stunning detail in some Suonatore fossils, and it was arranged in a manner that resembles the cardiovascular system of living mandibulates. The head anatomy of Suonatores too appears to line up with that of mandibulates, bearing precursors to the mouth apparatus that typifies the modern members of this group. And nor were the Suonatores the only basal mandibulates known from Cambrian sediments. Another such example is the 20-centimeter long Odoria, fossils of which show unambiguous evidence of large and well-developed mandibles. so it's clear that even as far back as the Cambrian, there was a wealth of mandibulate diversity. And though many of these ancient oddities didn't bear much of a resemblance to their modern relatives, they nonetheless show that the basic blueprint of all living mandibulates, including the centipedes, was already well established in this very early period. Within the mandibulates, centipedes belong to a clade called the Myriapods, along with millipedes and two other much more obscure groups, the Pauropods and Symphylans. A variety of myriapods are known from the fossil record. By far the most famous is the giant Arthroplera armata, a relative of the millipedes that could reach lengths of over 2.5 meters, and currently holds the prestige of being the biggest land arthropod known to have ever lived. Another fossil myriapod of worthy note is Pneumodesmus pneumoni, a roughly 430 million-year-old millipede whose remains include the oldest known spiracles, minute pores that many arthropods use to breathe air, and is thus perhaps the most ancient example of a land-living, air-breathing animal yet discovered. But fascinating as some of these early myriapods are, their fossil record is, on the whole, rather scant. Myriapods usually occupy moist, humid habitats, environments where decomposers like bacteria and fungi are prolific, so when they die, chances are their remains will be broken down and recycled by this ever-present cleanup crew long before fossilization can take place. In the case of centipedes, there's an additional factor. Their exoskeletons are relatively thin, unmineralized, and delicate, making their remains even less likely to be preserved. The limitations of the myriapod fossil record mean that retracing their evolution based off their fossils alone is a difficult endeavor. Luckily, there is a way to look beyond this limited selection of geological snapshots. It's called the molecular clock. Certain portions of an organism's DNA change at steady and consistent rates over time, and when we examine the diversity of modern taxa and the genetic differences between them, we can apply these rates of change to extrapolate backwards. The more difference there are between species, the longer it must have taken for those differences to accumulate, and the further back in time their diversification must have begun. Using this method, we can effectively reconstruct the myriapod's evolutionary history in reverse, painting a more complete picture than what can be gleaned from the fossil record alone. Multiple molecular clock analyses have been conducted on myriapods, and they indicate that these animals go back well before the age of their oldest known fossils, splitting off from their closest relatives sometime in the Cambrian. This presents us with a seemingly vast chasm in the fossil record. If this divergence occurred so deep in time, we'd expect to find early myriapod relatives in Cambrian sediments. Sure, there was a wealth of primitive mandibulates, but for a time there didn't appear to be anything that could be seen as adjacent to the ancestry of myriapods specifically. That, however, was set to change. Not, as you might think, with the discovery of some new group of ancient arthropods previously unknown to science, but with renewed interpretation of some already known arthropods that were, until quite recently, of controversial affinity. Meet the Euthycarcinoids, a rather tenacious clade of arthropods with a fossil record spanning from the Cambrian period all the way to the Triassic. The position of Euthycarcinoids in the arthropod family tree has long been somewhat ambiguous. Over the decades, a number of different affinities have been proposed. They've been interpreted as close relatives of crustaceans, of trilobites, or even horseshoe crabs. But our most up-to-date and complete understanding of these animals provides a compelling case that Euthycarcinoids were most closely aligned with the myriapods. A detailed examination of the remains of a Euthycarcinoid called Heterocrania, which included the best preserved head anatomy found in any Euthycarcinoid fossils thus far, provided novel insight into these ancient critters, and uncovered a suite of key traits that point to a close relationship with the myriapods. These, namely, included numerous features of the mouthparts, as well as the manner in which the legs attach to the body, all of which parallel the anatomy of myriapods. With this recent classification of Euthycarcinoids as part of the myriapod stem group, and therefore adjacent to the ancestors of true myriapods, these animals now fill the gap that once clouded our understanding of the early stages of myriapod evolution. and there is also evidence that even at the very beginning of their tenure, the Euthycarcinoids were flexing their many legs and clambering ashore. Fossil trackways matching the gait of these arthropods have been found in coastal sediments dating back to the Cambrian, and further evidence of the Euthycarcinoids' ability to venture onto land lies in their anatomy itself. The remains of another Euthycarcinoid called Euthycarcinoids show the animal possessed a pair of cavities or chambers toward the rear of its body, resembling the chambers that enclose the book lungs of arachnids, or the branchial chambers used by various crabs that spend a significant amount of time out of the water. This, in addition to the aforementioned trackways, gives ample reason to suspect that, though perhaps not fully adapted for life on land, Euthycarcinoids were at the very least amphibious, patrolling the shorelines and tidal flats of the Cambrian in much the same way that these soldier crabs are doing today. It would be difficult for animals to cut their ties with the water completely until the land became green, and eventually, of course, it did. Simple plants like mosses and liverworts began to clothe the earth. Then came the tracheophytes, plants with stems and roots. Now, with the greening of the earth, the animals could make their big move, and while the Euthycarcinoids may have scuttled across the coast, the Myriapods were set to press their conquest further afield, and establish themselves as arguably the first true land-living animals in the planet's history. And it wasn't long after the continents finally went green that the centipedes crept their way into the fossil record. The oldest definitive fossil centipede is Craterostigmomorphs crassulatum, whose remains date back to around 420 million years ago, during a period called the Silurian. Though fragmentary, it's still preserved in enough detail to tell us not only that it was a centipede, but more specifically what type of centipede it was. Craterostigmomorphs appears to have been an early member of a group of centipedes called the Suonatore, and their appearance need not be left to the imagination, for the Suonatore remain widespread and successful to this very day. These lightweight and exceedingly swift predators possess certain traits not seen in any other living centipedes. Most noticeably, they're the only centipedes with large, compound eyes, indicating well-developed vision, a feature that the rest of the centipedes, perhaps as an adaptation for a more subterranean life, have reduced or lost entirely. In addition to this, their spiracles, the pores through which they breathe, are uniquely among centipedes situated in a single row along the animal's back. Craterostigmomorphs, as a more primitive member of the Suonatore, did not precisely mirror the anatomy of its modern-day brethren. Its legs, though similar in many respects to those of living Suonatore, were considerably shorter. Moving forward in the fossil record, we eventually come across species of more familiar appearance. Latzelia lived in the late Carboniferous a little over 300 million years ago, and it very closely resembles the Suonatore of the present day. Younger still is the 115 million year old Fulminocursor, which again matches the anatomy of living Suonatore in just about every discernible way. Suonatore are one of 5 living orders of centipede, and determining how these 5 orders relate to one another is key to unraveling the progression of the centipede's evolution. Molecular studies have consistently reconstructed the centipede family tree as follows. Here the Suonatore are shown to have split off early on in the centipede's evolution, before the diversification of the remaining 4 groups. This, of course, lines up with the fossil record, with the aforementioned Craterostigmomorphs, a basal Suonatore, being the oldest fossil centipede known. The remaining 4 centipede orders together form a clade called the Pleurostigmomorpha, united by the placement of their spiracles. Unlike the Suonatore with their single row of dorsal spiracles, the Pleurostigmomorphs possess paired spiracles situated not along the top of the body, but on either side. These spiracles are not only placed differently, but also more complex than those of Suonatore. The Pleurostigmomorphs in turn can be split into 2 branches, the Lithobiomorpha and the Phylactometria. The Lithobiomorphs are a species-rich group of small and short-bodied centipedes, found worldwide in a variety of habitats from gardens to caves. These are considered the most primitive of the Pleurostigmomorphs, and share certain characteristics with the even more ancient Suonatore. One is their parental care, or should I say lack thereof. Like the Suonatore and unlike the remaining centipede orders, lithobiomorphs take the turtle's approach to parenting, depositing their eggs in the soil and departing without so much as a backward glance. The growth patterns of lithobiomorphs, too, align with those of Suonatore, with individuals of both these orders commencing life with a relatively small amount of body segments, and as the animal grows, the number of segments, and correspondingly the number of legs, progressively increases until the centipede reaches adulthood. This growth pattern is called anamorphosis. The other subdivision of the Pleurostigmomorpha, the Phylactometria, encompasses the remaining 3 living orders of centipede, and an additional extinct order that contains the oldest known member of this group, Devonibius, a minuscule creature less than a centimetre long, whose remains date back to the Devonian period about 375 million years ago. The Phylactometria, unlike the Suonatore and lithobiomorphs, exhibit a rather extensive degree of parental care, with mother centipedes brooding and nursing their eggs for the entire duration of their development, and continuing to guard their newborn babies until they're eventually capable of fending for themselves. Within the Phylactometria we have yet another division, with the group being split into 2 smaller clades called the Craterostigmomorpha and the Epimorpha. Craterostigmomorphs today comprise only 2 described species, one from Tasmania and the other from New Zealand. These rare and little-known centipedes retain some similarities with the Suonatore and lithobiomorphs. Like both of these earlier groups, Craterostigmomorph centipedes possess a total of 15 body segments when mature, and they too exhibit anamorphic growth where the number of segments increases over time, but not to the same extent. While the growth patterns of the Suonatore and lithobiomorphs involves multiple successive increases in segment count over the course of the animal's life, in Craterostigmomorphs there's only one. They start out with 12 body segments, and upon their first molt that number increases to 15, which is how it remains for the rest of the centipede's life. This partial anamorphosis, so to speak, can be seen as a transitional step between the multi-phased anamorphosis of the more primitive centipedes and the growth pattern of the next clade we're about to examine, the epimorph centipedes. In contrast to any of the centipedes we've covered so far, members of the Epimorpha exhibit no semblance of anamorphic growth at all, instead possessing the same number of segments and the same number of legs for the entire duration of their lives. The Epimorpha comprises the 2 most recently diverged centipede orders, the Scolopendromorpha and the Geophilomorpha. Between these 2, the Scolopendromorphs almost certainly need the least amount of introduction, for these include the biggest, the most venomous, and for many the most intimidating of all centipedes. And as far as we know, the Scolopendromorphs, now in the present day, are the most spectacular they have ever been, completely and utterly dwarfing any prehistoric centipedes yet discovered. And just a reminder, because I can already hear the agitated typing, Arthropleura was not a centipede. A narrow but notable selection of Scolopendromorph centipedes are known from the fossil record, the most ancient of which is the roughly 310 million-year-old Mesoscolopendra, which already bears a strong resemblance to living Scolopendromorph species. 2 more Scolopendromorphs of somewhat younger age, both dating to the Permian period, have been found in Russia. One is Permocrassicus, which was noted to resemble an intermediate state between 2 living Scolopendromorph families, the Scolopendridae and the Plutoniumidae, and may thus be of close affinity to the common ancestor between these 2 groups. The other is Permocryptops, which bears a strong resemblance to another modern Scolopendromorph family, the Scolopocriptopidae. Jumping forward to the Cretaceous period, we have Crateroricus, which lived alongside the Suonatore Fulminocursor and exhibited key features that identify it as part of the living family Scolopendridae. The other epimorph order, the Geophilomorphs, go largely overlooked, and they could be described as the centipede's silent success story. They're the most diverse of the 5 centipede orders, and occur almost worldwide, yet you may go your entire life without one ever crossing your gaze, for these are denizens of the underworld, highly specialized for subterranean life, with short legs and elongated, worm-like bodies, which allow them to cruise seamlessly through the underworld and squeeze into tiny holes more effectively than a Catholic priest.
The earliest fossil evidence of geophilomorph centipedes dates back to the Jurassic period, in the form of a species called Eogeophilus jurassicus. The remains of Eogeophilus, however, are not preserved in the finest of detail, and while it's readily identifiable as a geophilomorph of some sort or another, its less than optimal preservation makes it difficult to classify with any further degree of specificity. The period immediately following the Jurassic, the Cretaceous, holds some more examples of ancient geophilomorphs, and some are far better preserved than the likes of Eogeophilus. These notably include a quartet of specimens found encased in 98-million-year-old amber. The anatomy of these Cretaceous geophilomorphs was found to be very similar to that of their present-day kindred, indicating that geophilomorphs of more or less modern appearance had already become well established by this time.
It's one thing to talk about the timescale of the centipede's diversification, that is, the big picture after all, but no discussion about centipede evolution could possibly go without touching upon their trademark weapon, a formidable set of claws just behind the head that deliver a venom that in some cases can be a most virulent concoction. These venom claws, known as forcipules, or sometimes alternatively as maxillipeds or toxicognaths, are a weapon unique to centipedes, and the evolution of these structures and the venom they administer is every bit as fascinating as the evolution of the centipedes as a whole.
Forcipules, though often referred to as fangs or mandibles in colloquial speech, are in fact an entirely distinct set of structures. Though they resemble a set of mouthparts at first glance, these claws are not part of the mouth. Indeed, they're not even part of the head, instead being attached to the foremost body segment behind the head. This also means that a centipede envenomation, which is frequently described as a bite, isn't really a bite at all, since it's not being administered by a set of mouthparts. Instead, it could be more aptly described as a venomous pinch. Meanwhile, the centipede's actual mouthparts are comparatively small, weak, and non-venomous.
Similarly to the fangs of spiders or the sting of a scorpion, a centipede's forcipules deliver venom through a tiny hole situated close to the tip of the structure. This hole opens into a narrow internal passage called a duct that runs partway along the length of the forcipule. In Scutagiromorphs and lithobiomorphs, this venom duct is rather short, but in Scolopendromorphs it's considerably longer. The duct is divided into 2 distinct regions, the smooth duct, which is a simple tube, and the calyx, which is lined with glands that secrete venom. But how did this internal structure originate? Well, a 2014 publication has put forward one potential explanation.
Here, the authors proposed that this venom duct originated from glands situated on the outer surface of an ancestral centipede's limbs, that were then progressively internalized over time. In their initial stage, these external glands may have secreted noxious fluids harmful to prey and predators. Then, a possible first step in their evolution towards becoming venom-injecting devices would be the formation of a shallow groove down the edge of the forcipule, along which the fluid could be channeled toward the target, and if this groove were to become deeper, the flow of the toxins could be guided with greater precision. Then, a complete enclosing of this groove could form the tubular venom duct we see in the forcipules of centipedes today. The authors cited a few lines of evidence to support this proposal. One is the structural similarities between the internal venom glands of centipedes and the external fluid-secreting glands present in other myriapods like millipedes. In addition to this, it's been found that when centipedes shed their outer exoskeleton to grow, the entire venom duct is shed as well, further indicating that this now internal structure originated on the animal's outer surface. The venom duct of centipedes was also found to have a crest of exoskeleton running along its length, which may have been formed when the outer exoskeleton fused together to surround and enclose the duct.
From comparisons with other myriapods, it's apparent that forcipules are of distinct origin. They are modified legs, the only known example in the entire animal kingdom of a venom-injecting apparatus being derived from what was once a set of walking legs. And the progression of how these structures evolved is visible amid the diversity of centipedes today. A centipede's forcipules still exhibit the same basic structure as the animal's legs, consisting of a number of individual segments tipped with a sharp claw. And across the centipede family tree, we can see how this space plan has been transformed and specialized to varying degrees.
The forcipules of Suonatore are our best window into the earlier stages of their evolution, which shouldn't really come as a surprise, since Suonatore, being the most primitive of the 5 living centipede orders, are likely to retain more ancestral traits. Suonatore possess forcipules that are much more slender and overall leg-like than those of other centipedes. Thanks to their less modified, leg-like anatomy, the forcipules of Suonatore have a considerable degree of mobility, though their thin, delicate build makes them less effective at gripping and restraining prey. Moving along the centipede family tree, we can see further changes in forcipule anatomy. One concerns the coxa, the section at the base of a centipede's limb. In Suonatore, the coxae on each forcipule remain separate from one another, but in the Pleurostigmomorphs, the clade that comprises every other centipede order, they've been fused together into a single plate. In Lithobiomorphs, Craterostigmomorphs, and Scolopendromorphs, there is still a faint groove that marks this point of fusion, and in Geophilomorphs, this groove has faded out entirely. This fusion at the base of the forcipules hampers their mobility, largely restricting their movement to the horizontal plane, though this limitation is somewhat compensated for by the greater degree of physical power afforded by their more robust and reinforced build. Another evolutionary novelty seen in the forcipules of Pleurostigmomorph centipedes is the presence of a sharp cutting edge along the inner curve of the claw that allows these centipedes to effectively slice open and masticate their prey, something that Suonatore, with their primitive leg-like forcipules, appear to be incapable of.
But perhaps the most fascinating aspect of centipede evolution is not the development of the means to inject venom, but the venomous cocktail itself. Research on centipede venom has long been skewed toward one particular group, the Scolopendromorphs. These, being the only centipedes whose envenomations have the potential to be of medical concern, have been the principal focus of much of the research conducted on centipede venom, leaving the full picture of centipede toxicology incomplete. And recent research spanning all 5 centipede orders has found that the evolution of their venom is more complex, unique, and intriguing than one could have ever foreseen.
Analysis of venom samples collected from an assortment of species belonging to each of the 5 orders uncovered a huge degree of variety in their venom's composition. Across the board, a total of 93 different types or families of proteins and peptides, the basic components of venom, were identified, and of these, between 66 and 68 were exclusive to a particular centipede order. Conversely, not a single one was found to be present across all 5 orders. This indicates that across the time scale of the centipede's history, the composition of their venom was extraordinarily dynamic, with numerous instances of venom components being both gained and in some cases lost over the course of their evolution. And analyzing the genetics that underpin each of the proteins and peptides that make up the centipede's venomous cocktails allowed the researchers to gauge when each of these individual components arose during the centipede's evolution, and the results were truly fascinating. It was found that the ancestral centipede's venom was a very simple concoction, consisting of only 4 toxin families, a far cry from the nearly 50 identified in some living centipedes. It also appears that the centipede's venom remained relatively basic for quite some time afterwards, with only a couple extra components being acquired during the earlier stages of the centipede's evolution. And it wasn't until after the centipedes had diversified into the 5 separate orders we know today that their venoms underwent a more drastic increase in complexity, with each order independently evolving their own sophisticated venomous arsenal.
Venom typically originates via the modification and weaponization of proteins and peptides that are already produced by the animal for another purpose, and for the most part this does appear to have been the case for centipede venom, with the majority of toxin families having a similar genetic basis to those of many other animals. However, some of the centipede's venom components appear to have no equivalent in the entire animal kingdom, a sign that these toxins may have been of much more unique origin.
A 2021 study found evidence that some of the genes that encode certain components within centipede venom originated from external sources including bacteria and fungi, and were recruited via a process known as horizontal gene transfer, where one organism takes up genetic material from another without reproduction, as opposed to the much more familiar concept of vertical gene transfer, where genetic material is passed generationally from parent to offspring. At least 5 of the venom-encoding genes possessed by centipedes appear to have been acquired in this way, and 3 of these, all of bacterial origin, are, in their original host, responsible for what are known as virulence factors. Virulence factors are what cause certain strains of bacteria to be infectious and harmful, and centipedes, by recruiting the associated genes, have incorporated these bacterial weapons into their own arsenal.
Centipedes, to me, are a treasure trove of fascination. At the surface level, the mere sight of a centipede has always captivated me, even if I didn't initially regard them with the same level of fondness as I do now. And then, when you probe beneath the surface, each layer you pull back reveals yet more to marvel at, from their unfathomably deep history to the remarkable intricacies of their venom. Whatever your feelings about centipedes may be, I can only hope that this video can, at the very least, give you reason to appreciate these awe-inspiring predators just a little more.
Centipedes aren't the only inhabitants of the undergrowth with a deep and fascinating history. Spiders, too, have an incredible story to tell, and if you want to learn all about their evolution, then head on over to this video here. And don't forget to subscribe so you don't miss out on any future content. Thanks for watching, and I'll see you on my next adventure.